Good afternoon, everyone. Hello, welcome. Really pleased to be here. It's been a really nice few days in Shanghai. It's my first time visiting and really excited to be presenting today on our work at the Doherty Institute in Melbourne, Australia. So for those that don't know, the Doherty Institute is one of Australia's leading infectious disease research institutes. It's a joint venture between one of our hospitals, the Royal Melbourne Hospital, and our University of Melbourne, so one of our premier research universities. So let me just click through. Down. Sorry. Great. So this is a quick overview of what I'll be presenting on. So I'm from the infectious disease side of the world and presenting a bit of background in what we're looking for in both diagnostics and surveillance tools in infectious diseases. And then stepping through a few examples where we're looking at using Twist and other companies' products for culture-independent sequencing as part of public health microbiology. So this is a really nice kind of intro slide as to why we need to use culture-independent methods for sequencing. If you go back over maybe 2 or 3 decades, everything in public health microbiology was based on growing up bacteria or viruses to then have enough material to sequence them and say what's happening in their genomes or what's happening in their biology. But as things are increasing in pace and we need to have answers really quickly in the context of public health and even in clinical microbiology, having culture-independent methods that we don't need to use culture for, is becoming really, really important. And so you might have heard this described as metagenomics, is another kind of phrase that represents sequencing everything in a sample rather than growing something out of the sample and sequencing it on its own. Yeah. Sorry. Is this better? Right up here. Okay, sorry. Thank you. And so these are our frontline tests, things that we routinely use in public health microbiology to either identify a pathogen or to characterize a pathogen. So you might be familiar with antibody-based tests or serology, which is that top line, where you're looking for individual's immune response. So the immune system of a person responding to a pathogen. So that's really important still in a lot of different diagnostic contexts. But more recently, we're using other molecular techniques shown down the bottom as an example, where we're looking for pathogen nucleic acids. Thank you. Testing again. Is that better? Great. Okay, cool. Thank you. And so this bottom line is becoming increasingly important, and through COVID was really, really central to our response to identify patients that had COVID infection. Well, I can hear that now. And so what we are looking to do at the Doherty Institute is to use nucleic acid-based tests, so things in line with the bottom here, to characterize pathogens and to detect them in different or more useful ways. So I'll outline a few of those now. As an example, if you're looking at someone that has respiratory disease, so say cough or symptoms of fever and things that you might now associate with COVID, that could be caused by a number of different viruses, things like influenza, respiratory viruses, coronaviruses, but other types as well. It could also be caused by other pathogens that aren't viruses. So lots of pneumonias are caused by bacteria. Some rare pneumonias are caused by plasmodia, and you could even have a non-infectious pneumonia. So someone that doesn't necessarily have a pathogen, but has, you know, an autoimmune condition or something similar that is giving you the same symptoms. And so what we're aiming to do is using a culture-independent approach to be able to say which of these is the right answer really quickly, as that can inform treatment and make sure that those patients are able to access care. And so what we've been using are panel-based sequencing approaches. So these are things that, like Twist might offer, where you are designing a panel. In this case, Twist has a number of pre-made panels where you're able to pull out bits of RNA or DNA from a sample, depending on how you've designed it, and sequence those specifically. So in the case of viruses, what we can use is a product called the Comprehensive Viral Research Panel from Twist, and that's able to enrich a number of human viruses, effectively all known human viruses, from a sample. So if there was at the end of your sequencing run, maybe 1 or 2 reads, you'd expect that to increase 5,000-fold by adding in an enrichment using the Twist Bioscience comprehensive panel. So really, really useful, really important to increase the sensitivity and to make sure that you're able to get all the information you need out of your test. This is a quick overview for those that might not be familiar on the panel enrichment approach. So your generic workflow is that you would make a library shown on the left, like you would use for any other sequencing protocol, and you're adding a step where you're enriching out parts of that library that you want to selectively sequence, things that you really want to have more information on. And this would be your viruses or other pathogens of interest that you're trying to pull out of your sample. So really, really useful, and we don't necessarily need to know what viruses is in your sample to be able to pull it out using a product like the comprehensive viral panel. And so this gives you a lot of information. This is just a quick overview slide, but it can tell you a number of bits of information that are clinically actionable or actionable on a public health level. So how related samples might be, if there are things like outbreaks, if an individual might have co-infections, which are quite often hard to identify using routine diagnostics, and other bits of information about the genotype of that pathogen to do with antiviral resistance or antibiotic resistance, things that can really guide therapy. This is another few slides just on the particular panel that I'll be presenting some data on, the Comprehensive Viral Research Panel. So this includes more than 1 million probes, more than 1 million bits of DNA that can be used to pull out selected sequences, in this case, human viruses, as well as a number of bat coronaviruses and other things that could be potential zoonoses, so things that could spill over into humans and cause disease. So really, really comprehensive, as the name would suggest. The other really useful thing about this panel and the panel-based enrichment approaches more broadly is that they don't need to be an exact match to something in your sample to be able to pull it out. It's based on hybridization, so if your DNA is close enough, within say 10% sequence identity to what's in your sample, it will hybridize to it and be able to enrich that out. So it can be used for novel viruses, things that you might not have seen before, and we've used this already for things like Japanese encephalitis virus and monkeypox and things that we didn't kind of design to be in the panel and to have an exact match for, but were already in the panel as they're known human viruses. And so this is some new data from our team, which is looking at respiratory viruses. So as a public health reference lab based at the Doherty, we have a number of diagnostic tests which we run on respiratory viruses, so things that you might be familiar with as influenza or RSV, even COVID. And so if we could have one test that can diagnose which respiratory virus a patient has and allow us to sequence it and characterize it, that would be really useful. And so what we're showing here is that we can do that using the Twist Comprehensive Viral Panel. We don't necessarily need to know anything about the sample other than that it's from a patient that has symptoms. And as you can see, there's a relationship where the higher the load of the pathogen, as shown by the PCR results down the bottom, the better results you get, as you'd imagine. If a patient has really bad symptoms and a lot of virus, we get really, really good genotypes. And as it gets rarer and rarer, fainter and fainter amounts of virus, then it becomes harder and harder to detect and to characterize. So really promising, and we can show that stratified or broken down by different viruses. So these are common respiratory viruses that we are interested in from our public health work. And again, showing that trend where as you've got high loads of virus, you get really good sequencing results. But the main take home from this is that we can characterize any of these respiratory viruses on a genome level. We can then use that to say subtype things like influenza or RSV, which is important for vaccine selection or vaccine design. We have a number of companies that are working with us looking at would their new vaccines be able to cover the kind of diversity of RSV or flus that are out there based on this type of information. So really promising. And what we've also seen is that this approach has a lot of advantages over sequencing methods that you might be familiar with through COVID, so things like your amplicon-based methods. So here, this is an example of an amplicon scheme where you're effectively doing lots of PCRs, potentially hundreds or thousands of PCRs, to amplify up a genome of a pathogen, which worked really well for SARS-CoV-2 or COVID-19 because it was an outbreak. Everything was the same. It hadn't had any time for it to to change and to diversify. But this approach doesn't work well over time. Say for COVID now, where we have a number of different variants, a number of different types of COVID, the amplicon schemes might drop out as sequence changes. You might have primers that no longer amplify a region, and quite often those are really important regions, things like the spike gene in COVID, which is diversifying at a greater rate as it's trying to escape the vaccine-based immunity. So why is a panel-based approach better than one of these amplicon approaches? And it's because it can tolerate that diversity a lot better. As I mentioned, it can tolerate 10, maybe even 15% sequence diversity before it will stop annealing and stop being able to enrich those sequences. So really promising, really useful for kind of rock solid, maintained genotyping and detection of pathogens. So this is a summary slide, just going through that work. It shows real promise for our work in responding to viruses of both respiratory pathogens, but also pandemic potential pathogens, sorry. And we had a paper out just last week looking at our monkeypox sequencing, so mpox, which you might have seen in 2022. We actually used the comprehensive viral panel for our frontline testing for monkeypox. Just took it off the shelf and it worked first time for a number of our samples. So if you're interested in that, please come and say hello afterwards, and I'm happy to speak more. The last few slides will be focusing more on uncultivable organisms. So what I mean by that is the respiratory viruses or SARS-CoV-2 can all be grown in a lab. They can be amplified up in routine cell culture to study or to sequence. But what would be really useful is sequencing things that we can't do that for, things that are uncultivable organisms, either because they're from environmental samples or because we don't have cell culture systems for certain organisms. Not everything can be grown in a lab. So a few examples of those using culture-independent sequencing here. So a few years ago, we had a research project looking at an organism called Treponema pallidum. And this is a bacteria that causes syphilis, so a sexually transmitted disease that has some very severe sequelae. And what we're seeing is that in Australia, it was really increasing in incidence over the last decade, which led us to kind of question, is this being driven by a new form of this bacteria, a new variant? Is it, an outbreak of something that we really need to respond to specifically? Or is it changes in public health? Is it, people behaving differently and transmitting the bacteria in different ways? And we could answer that using genomic methods. And so this is using, again, a panel-based culture-independent sequencing method to enrich material out of these samples directly and sequence them. As you can see, we have that trend where the higher the amount of pathogen, the better sequencing we get. And as it gets fainter and fainter, we get less, but still really, really good. And some of this work is published in Nature Microbiology as part of a global consortia effort. But we've also got our standalone paper, which is published in Lancet Microbe, which is looking at our Australian diversity of Treponema pallidum. And the take-homes from this is that we don't have an outbreak, we don't have a new lineage in Australia, we have everything that's out there globally, we have a lot of things that we are importing from other countries, and so it's probably a public health-based increase in incidence. It's not something about this pathogen that has changed that's potentially problematic. And then lastly, this is some more recent work which is yet to be peer-reviewed, but we have an outbreak in Victoria of an organism called Mycobacterium ulcerans. So this is a bacteria that can live on possums and somehow was affecting humans. And humans don't usually interact with possums in Victoria. They're an endangered species, and so we were kind of trying to figure out how these two were related as we see outbreaks in humans and possums at the same time. We were thinking maybe there's a shared exposure, maybe this bacteria is in the water or is carried by another animal that we haven't yet sampled. But what we were able to show using culture-independent sequencing, again a panel-based method, is that there's a link between mosquitoes and possums. So people that have a mosquito bite can have Mycobacterium ulcerans transmitted from when the mosquito bit a possum previously as kind of an intermediate vector. And so able to prove that really conclusively. And as you'd imagine, the genomes of the bacteria found in a mosquito and a possum and a human patient were almost identical, which we're able to show on this little figure here, where things from possums and mosquitoes all fit into this phylogenetic tree, which is based on the human bacterial sequences. So really, really, like, for me, exciting work. The kind of one sentence take home is that one of these samples was just a single mosquito, and we could prepare that sequence from that and get a complete Mycobacterium ulcerans genome, so the bacteria that's carried by that mosquito, from that one mosquito. So really powerful technology. And so that's a quick summary of the work that I've presented. I think you'll imagine that this is a really powerful tool for public health. It offers really streamlined approaches, as we can effectively take things off the shelf to use them straight away in the context of an emerging pathogen or an outbreak. We can also tailor them to target, in our case, very niche organisms to do with possums or to do with Treponema pallidum. So you can kind of fit anywhere between comprehensive and very focused research questions or public health questions. And what this has led to is a number of public health interventions. So we're able to have relationships with our Department of Health, our kind of government, to say this is an outbreak or this isn't an outbreak and this is how you should respond to it accordingly. So really useful from their point of view. And the last slide I'd like to touch on is that this is all very what I would call the kind of pointy end of public health, where we're interacting with the Department of Health or a government body. But these culture-independent methods are also really useful in the context of like fundamental research as well that could inform public health down the line. So these are two papers that we've been involved in, both published in Immunity, which are looking at different SARS-CoV-2 variants, which we didn't use culture for, we just use culture-independent methods. And both of these papers are around kind of guiding how vaccines are going to work long-term for COVID. So really useful, not just for the very pointy end of public health, but useful throughout the entire kind of developmental pipeline. So we'd just like to say thank you again. I'm really pleased to be here, really welcomed, and yeah, I'm happy to take any questions.